This is a video tutorial for BugNet where I will briefly explain how to select species and individuals to take damage incidence and damage percentage measurements in each plot.
Here is an example plot. This is a herbaceous plot in Switzerland. I have already done the cover estimate for all the species in the plot. And using this cover estimate, I found that 2 species in this plot make up 80% of the plant cover. This doesn't include bare ground or rocks or overstory species. Select up to 5 species in each plot. These species will be the ones with the highest cover in the plot, and their combined cover makes up to 80% of the vegetation cover in the plot. The first species in this plot that makes up the highest amount of plant cover is called Nortia arvensis. It's the one with the bright green lobed leaves. The second is a Veronica species. It's the one with small leaves and little purple flowers. For the Nortia species with the highest amount of cover in the plot, there were 9 individuals in total. This meant that I measured the incidence of damage on every individual in the plot. Don't forget to measure incidence of damage for both herbivory and pathogen damage. And each one of these is split into its separate groups. For example, mining or galling herbivory, and also pathogens such as rusts or powdery mildews are separate. Once I had measured incidence of damage, I then had to select a random subsample of only 5 plants to take back to the lab to look at percentage damage measurements. To randomly select this subsample of 5 plants from the 9 plants in my plot, I simply used a random number generating app on my phone. Before I took these plants out to take them back to the lab, I measured plant height on all 5 plants. Once you take these plants back to the lab, you'll measure percentage of herbivory and also pathogen damage. Likewise, you'll measure some plant traits like SLA and LDMC. The Veronica species was a little more difficult. There wasn't a clear separation of individuals within the plot. It was more of a spreading herb. So I divided it into units, each one with its own flower. Obviously, there were more than 20 units in this plot, so I used the quadrant method. I divided the plot up into 4 quadrants of equal area. Then I haphazardly, randomly selected 10 units of Veronica, proportionate to how many were in each quadrant. This can be a coarse randomization of selecting the individuals, but it still needs to be random. I then numbered these individuals from 1 to 10, and again, using a random number generator app on my phone, selected just 5 to take back to the lab for damage percentage measurements and leaf trait measurements.
We've now seen 2 different examples of ways to select individuals within a species in the plots. We saw when there were 10 or less individuals, we selected all of the plants to take damage incidence measurements on. We also saw when there were more than 20 individuals, we did the quadrant method to pick 10 individuals to measure damage incidence on. Now we're going to see another example where there are between 11 to 20 individuals.
The Gallium species was one of the species that made up the 80% of plant cover in this plot. There were 12 individuals of Gallium in this plot. In order to choose just 10 individuals to take damage incidence measurements on, I labelled each of the 12 individuals with numbers 1 to 12, and then again used a random number generator app on my phone to pick just 10 to measure damage incidence on. I used the same random number generator to then select 5 of those 10 individuals to take out of the field and back to the lab for percentage damage measurements and for leaf trait measurements. Remember that if your individuals are really damaged, take extra ones back to the lab, because the leaf trait measurements like SLA and LDMC must be measured on undamaged leaves.
Thanks for watching. If you need any more information about these measurements or about BugNet, don't hesitate to visit our website at bugnet.org or contact one of us via email.